Thank you very much. Thank you to the organisers for inviting me to speak here today and to come and present neonatal data in an afternoon which has been so clearly focused on the life course. So the patient population we'll spend the next 20 minutes or so discussing is the preterm infant, an example of whom is shown here, and this is a member of one of our birth cohort study. So this is broadly defined as babies born with a birth weight of less than 2.5 kilos or 5.5 pounds. Across the UK, this accounts for 7 to 8% of all births. There's a countrywide variation that ranges from 5% in the best of European countries at this to 18%. And broadly speaking, the goals of care during this period are to maintain physiological homeostasis while the whole organism grows. And for the focus of our talk, we'll focus on the brain. And you can see right there that the head actually almost doubles in size for a baby born at 25 weeks over the following 2 to 3 months that they spend in the NICU. So the most informative data about how these children fare, at least in the UK, comes from the EPICure cohort, which was a cohort run over the whole of the UK and Ireland, whereby the investigators recruited every single baby born at less than 26 weeks during a 10-month period in 1995, and have followed them over the years. And this is their school-age outcome at the age of 6, where on the y-axis is cognitive score, which for the most was the Kaufman ABC, and on the x-axis is their gestational age week at birth, going from 23, 24, 25 weeks. And investigators had the great foresight to include a classroom comparison group. And what you can see is that at school age, this population of children are achieving well under the population norm, and that compared to the classroom controls. And in fact, more than 41% of them are performing more than 2 standard deviations below the expected. Now, this is a group of perinatal medicine has improved over the past 2 decades, and survival rates are increasing, but this particular problem has not, and is replicated in various cohorts that collect these data around the world. This translates into educational difficulty. So these are now data from Scotland collected by Jill Pell's group in Glasgow, where they were able to link the education record with the birth record and show a clear relationship between the need for special educational support in the classroom on the y-axis relative to gestational age at birth. And you can see that there's a need for extra support in the classroom right across the gestational age range. So it's not just the tiny babies that are having difficulty with this. It's right up to birth at 39 weeks. I won't go into all of the data from all of the studies, but there's general consensus that aspects of cognitive function and cognition that are altered in the preterm population of children and adults are the following. There are global and specific learning difficulties, particularly with numeric operations. There are difficulties with planning, inhibition, working memory, and verbal fluency. Inattentiveness without hyperactivity seems to be a problem. Anxiety is a problem. There are social difficulties, particularly with peer relations. And there's an increased likelihood of both screening positive and receiving a diagnosis of autism. And as the oldest cohorts have started to mature, it's clear that there's an increased burden of psychiatric disease in adulthood, at least measured by use of psychotropic medications and hospital admissions. And so there does seem to be this legacy across the life course from being born too soon. So I want to spend the next few slides discussing how we've used MRI to map the developing brain and map the alterations in brain development that take place in association with preterm birth. So these are images from a single subject whose mum gave me permission to scan the baby at several points over 15 weeks following birth at 25 weeks of gestation. And we got these, we've learned a tremendous amount from this very, very rapid period of brain growth over what is effectively a 3 to 4 month period. We've learnt that the brain volume increases linearly, but the complexity of the cortical surface increases exponentially during this period. And I and a number of other groups around the world have been keen to try and understand what the effect of this process taking place outside the womb is and in an incubator. And so we recruited a group of patients and took MRI at term equivalent age. So this is after their period of NICU care, just at the point when they're going home. And the image you see on the left there is from a completely healthy control, born at full term, imaged on day 2. And the image on your right is from a preterm infant born at 26 weeks, imaged at the equivalent age of 40 weeks of gestation. And there's consensus that the extracerebral spaces are a bit wider, but there's diffuse signal intensity changes in the white matter. So on T2 weighted imaging, the white matter looks brighter, and this is seen in about half of preterm infants at term. And because it occurs with a similar sort of prevalence to the later difficulties, it's raised a lot of interest as a possible substrate for later impairment. Now, for those who are a bit worried about the subjective judgment of defining diffuse high signal intensity on white matter, this is measurable using diffusion magnetic resonance imaging. And for the non-imagers, this is essentially an imaging technology whereby the gradients of the MRI scanner are sensitized to the molecular motion of water, and it's water therefore the MR scanner picks up the signal from water and transforms it into an image. And so water behaves very differently in different tissues. And so here you see a green outline of a major white matter tract. And along in that area, water would tend to move with direction, because it's constrained by axonal fibers and myelin and so forth. And when that's modeled, therefore, by an ellipsoid, there is a directional purpose to the motion of water. However, if you take a diffusion scan of the CSF space here, outlined in red, water will tend to be moving with Brownian motion with no direction, and that's modeled as a sphere. The MR scanner converts that into an image, and so areas that are bright show where water is moving with a sense of with direction rather, and that therefore highlights the major white matter fasciculi, because these are where axons are tightly packed, myelination is taking place, and that is bright on a diffusion weighted image. There are two measures to quantify that. One is mean diffusivity, measured by, which is the unit of which is area covered per second, and fractional anisotropy. Simple measure ranging from 0 to 1, where 1 is water, would be a water molecule moving in a totally straight line, and 0 is would be moving with no direction. And from these, one can make inferences about the amount of brain water, axonal thickness, axonal density, and myelination. And therefore, they provide insights into underlying tissue microstructure, which is very helpful for quantifying this diffuse white matter injury that we see. So in the first study we did of this, we took a very simple experimental approach, which was to define regions of interest in the white matter and in the deep grey matter, and to measure these diffusion parameters. We found that the preterms who had the appearance of excessive signal intensity in the white matter have higher ADC values, higher mean diffusivity. The brain is more waterlogged compared to controls. But this was not so in the deep grey matter structures. Then wanted to ask, what is the consequence of that diffuse white matter injury on overall brain growth? And for this, I used a technique called deformation-based morphometry, whereby one transforms brains into a common template space and therefore can model differences in anatomy from those transformations. This is an example of two different individuals where the brain image on the left is going to transform over a period of 6 seconds until it resembles the person's on the right. I think it is. Sorry. So if you keep your eye focused in on the ventricular outline, you'll see it's gradually morphing until it resembles the individual on the right. And from, let's just run that again. Now, once that transformation has been computed, one can then go back to the transformations and ask of the transformation where are the spatial areas of increase and decrease in size in order to adopt the new anatomic space. And you can apply this, you can scale up. We applied this then to a whole group of preterms with and without the white matter disease. And these are just the results visualized in a single infant space, but they represent an atlas-based approach to saying what's the average difference in brain structure between two groups. And so we found that preterm infants had lower brain volumes in the centrum semiovale, the dorsomedial nucleus of the thalamus, which turns out to be a very fascinating nucleus, closely involved in a lot of the functions that this population of children have difficulties with later, and the globus pallidus. Whereas preterms who had normal white matter, as measured by ADC values, did not have abnormal growth. So this raised the possibility that this problem is a manifestation of disconnectivity, because it's abnormalities in the white matter that created these failures of brain growth in the deep grey matter, suggesting that those two, but without evidence of injury in the deep grey matter. We then went on, this was work done by a PhD student of mine named Gareth Ball, to use tractography to see to study thalamocortical pathways. And you can see here that for the preterms versus completely healthy controls, there were reductions in connectivity between four of the major thalamocortical pathways, indicating that this problem of preterm birth is one of disconnectivity. Now, the technology moves on at pace, and so this is work by Devasuda and Blagan in collaboration with Mark Bastin and Simon Cox, where Devasuda was able to look more widely than just the thalamocortical networks and to consider other major fasciculi, as shown here. So these are the white matter fasciculi segmented out of the neonatal brain. Now, it turns out that the variance of these diffusion parameters within these major white matter tracts is substantially shared across those white matter tracts, and in this way, the newborn brain is a complete facsimile of the adult brain, as shown by Professor Deary and other colleagues here a few years ago. Now, this is interesting because this is the predominantly unmyelinated brain, and yet the white matter architecture is a facsimile. And in collaboration with Simon Cox, we then went on to examine the extent to which preterm birth alters that general property of white matter structure, and indeed it does. So these are the grayed-in boxes are healthy controls, the preterms are the light boxes, and the coloured data represent the various diffusion parameters. And so preterm birth not only affects specific thalamocortical networks, but has a global effect across all white matter microstructure. So I want to show you two examples of how we've started to use these imaging to relate back to biological data to try and understand what the biological determinants of this altered connectivity are. And the first is inflammation. One of the most common causes of being born too soon, is if your mother develops a condition called Chorioamnionitis, which is essentially a bacterial infection of the membranes that surround the fetus. Usually it arises because there's an ascending bacterial infection from the vagina through the cervix, and it infects the membranes. And this creates an inflammatory response in the fetus, as measured by raised inflammatory cytokines in cord blood. And there's been uncertainty really in the field about what the extent of the fetus having an inflammatory existence in utero is on brain structure. So we have the hypothesis that if that a group of preterm infants who are exposed to intrauterine inflammation through chorioamnionitis would be associated with white matter injury. We collected a large group and got placental histology from all of them. This is just examples. This is the fetal membranes in here. Show the blue dots here are maternal inflammatory neutrophils that have gone into the cells because there's bacterial invasion of the membranes. And this is healthy amniotic membranes. Similarly, this is a section through an umbilical cord. These blue dots are actually fetal cells. As the fetus is mounting a response to inflammation, fetal neutrophils are ramping up and trying to exit through the umbilical cord into the amniotic fluid. So we were able to define, using these tissue definitions of in utero inflammation, a group who had any evidence of chorioamnionitis or infection of the cord, and a group who did not have that as a cause for their preterm birth. We scanned them on a 3 Tesla MRI system, and to cut a long story short, use a feed and wrap technique. There's various technologies for supporting young babies in scanners, but the one that we go for, that we find works, is to just feed the babies, time it with their sleep, swaddle them, have the mums involved, and we get good images. The image processing technique that we used is TBSS. So this it stands for tract-based spatial statistics. Effectively, it allows you to take the diffusion images from 2 or more participants, as shown here. In this case, it was a large group, divided into those with in utero inflammation and those without. You register the data into one common space so that you can compare like structures with like. That produces a mean for the entire study population. And then you can strip away voxels that are not of interest and just examine the white matter skeleton. And there are various reasons why this experimental technique is desirable, not least because it helps to control to some degree the multiple comparisons problem of multiple tests across many voxels. It just strips down to the bare minimum of the white matter skeleton. And we found that indeed in the babies that had exposure to chorioamnionitis, there were reductions in FA that were very diffuse, widespread throughout the white matter, shown here in blue, where yellow is the underlying white matter skeleton. And so clear evidence that even after adjustment for various other factors known to hinder brain white matter tract development, the preterm group, at the point that they're going home, 2 or 3 months after birth, if they had an exposure to inflammation in the womb, had lower FA throughout their white matter. So other speakers have touched on epigenetic modification, because we've now, of course, started to think about what mechanistically might mediate these effects between an adverse exposure and tissue injury. And Ricardo introduced the concept of DNA methylation, which is a dynamic process by which the genetic code, which is fixed, can be dynamically expressed depending on methylation of CPG sites. Now these following 2 slides are quite text heavy, but please bear with me while I build the case as to why this is a relevant thing to consider for the brain in early development. Now we know that through the fetal life, there's dynamic expression of DNA methyltransferases. These are the apparatus that enable DNA methylation to happen, and they gradually build up during fetal life. There's also a dynamic trajectory of the brain methylome across fetal life, tending towards hypomyelination. And it tends to be the case that as CPG sites get less myelinated, genes get more expressed. And this indeed is the case as the fetus progresses, as the fetal brain progresses through gestation. And of course, there's the classic example of Rett syndrome, which is a very severe neurodevelopmental disorder seen in girls who've got mutations in the MECP2 gene. And from preclinical studies, actually, there's dynamic regulation of DNA methylation associated with neuronal plasticity, memory formation, and circadian processes. And so these are happening at very rapid turnover as the brain develops. Now, there's also evidence from humans, fairly persuasive studies, that this particular mechanism of DNA methylation could mediate gene-environment interactions that link early life stressful events and adverse psychiatric outcomes in later life. And so in this particular study of people, suicide completers, this was a post-mortem study where the investigators showed changes in DNA methylation in conjunction with glucocorticoid receptor expression in the hippocampi in people who had completed suicide but with a prior history of severe child abuse, compared to those who commit suicide but have not had that early life exposure, suggesting that the early life event somehow reprograms the HPA axis. Similar result, that childhood maltreatment, classified as sexual or physical abuse, was associated with distinct epigenetic profiles in adults who went on to get post-traumatic stress disorder versus those who did not have that early life exposure. And finally, in the Helsinki birth cohort, men who were exposed to early life stress, which was forcible removal from their parents during the war, and then went on to develop depression, had altered DNA methylation profiles. So fairly compelling evidence that DNA methylation may well be one of the mechanisms that links early life difficulty with psychiatric disease. So we recognize that preterm birth is a very profound early life stressor physiologically, and would that be associated with alterations in the methylome? So we recruited preterm infants and controls. We ran the methylation 450 trip with Lee Murphy as a collaboration here. And we boiled down to 242 sites that were differentially methylated between preterm infants at term and completely healthy controls. There were 60 differentially methylated sites in protein coding regions and 25 in gene bodies, and of these, 10 were associated with neural function. These are they. There's a list of genes here that we could talk through at the end, or the reference is there, but essentially their functions were very interesting and plausible, and plausible biological mechanisms by which brain injury is known to take place in this population. So there was altered blood-brain barrier permeability to large amino acids important for nutrition, but also thyroid hormones. There was a suggestion, there were genes involved that encode excitotoxic mediator clearance, such as glutamate, quinolate, and this really sort of focuses attention on a number of different mechanisms that may well be important. So to conclude, preterm birth is associated with focal volumetric reduction in the thalamus and the globus, but also with disconnectivity of that network, but more generally other white matter fasciculi. The pathway to white matter injury begins in the womb for some babies, specifically those who've been born following chorioamnionitis. And that epigenomic profiling of preterm infants reveals DNA methylation differences at 10 loci that are known to be associated with neural function. And this perhaps suggests interesting genetic and epigenetic causes of brain injury, but also repair in this population. I just want to finish by saying what we're doing now, and that's in the form of the Their World Edinburgh Birth Cohort. Their World is the name of the charity that has substantially funded the cohort. We're going to recruit 500 babies. In fact, it's well underway, and collect very detailed information about anatomy from MRI, physiology, biology, socioeconomic factors as the children grow. There's ophthalmic measures, so this is for retinal nerve fiber layer thickness. So far, the charity or the funding sources, of which there's now more than one, have agreed to fund data collection up to 5 years, and these are the planned time points for assessment, but very much with an intention that they will continue to do so, so that we could see this group of children every 5 years until they grow to adulthood. And the goal really is to understand what the biological drivers of brain injury and repair are in the newborn period, to what extent events in childhood interact with the newborn events to determine life course outcomes, and to perhaps shed some new insights into early childhood factors that lead to adult neuropsychiatric diseases. So I'd just like to thank a number of people in my group who've contributed. The multidisciplinary effort that really is the Their World Edinburgh Birth Cohort, the funders, the Scientific Advisory Board, who've provided really very helpful steer over the past couple of years, and not least the families. Thank you very much.